Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the Histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below.
Hi and welcome back. In the last of the 3 Neurohistology lecture series, we'll integrate the neurohistology with the neuroanatomy. Enjoy! This is the fun part, where we get to integrate the neurohistology with the neuroanatomy. Because a lot of histological organizations actually show up at the anatomical level. Here's a figure of the brain anatomy. This is actually not a human brain, FYI. The right and left hemispheres are well appreciated, as well as the cerebellum. The right cerebral hemisphere has been cut in a transverse section. And here, at the anatomical level, we can see a number of different things. First, we can see that the outside layer of the cerebrum has a lot of these undulations and folds. And secondly, we can actually see that at the gross level, that the outside layer of the cerebral hemispheres are a little more darker than the inside layer. And another thing that we see is that within the cerebral hemisphere, we actually have a space which is called the ventricle. And this in real life would be filled with cerebrospinal fluid. And of course, at the histological level, we'd be expecting to see the ependymal cells lining this ventricular space. So at gross level and at histological level, the outer layer of the neural anatomical structures are called the cortex. And here in the cerebral hemispheres, the outer layer is also called the gray matter because it is physically gray or darker in color. At histological level, these areas are darker because they contain numerous cell bodies of the neurons. The inner layer of the brain that looks slightly lighter and kind of glistening in color is called the medulla. Now, while the cortex refers to the outside layer or a bark, as in a tree bark, medulla refers to the inner layer of any given tissue or an organ. We've actually encountered these regional terms, the cortex and medulla, when we learned about the bone anatomy. At any rate, medulla directly translates to the marrow. So here in the cerebral hemisphere, the medulla is comprised of this shiny yellowish white structure. So we call this the white matter. And this area looks white because these are all comprised of radiating axons that are richly myelinated by the oligodendrocytes, and myelin sheaths mainly being comprised of the phospholipid bilayer rich cell membranes. Of course, they would give off the whitish yellowish color. Next, we have this concept of nuclei. Now, we're not talking about the nuclei of the cells. We're actually talking about a collection of gray matter that are stuck within the medulla at the gross anatomical level. So these islands of gray matter within the deeper layer of the cerebrum would contain a lot of neuron cell bodies. Hence, they give off this gray color within the medulla. And then we have the tracts, which are these bundles of axons that are running in various directions within the medulla. Now then, if we were to look at a small area of the cerebrum under the microscope, we would still be able to see the anatomy of the cerebrum with the gray matter or cortex up there, with each one of these darkly staining dots representing the nuclei and the cell bodies of the neurons, and the white matter, which is comprised of collections of axons which radiate from the gray matter or the cortical layer. To confirm this, let's look at this tissue at a higher magnification, in which case we're able to see each one of these darkly staining structures being the cell bodies of the neurons, with actually pale staining nuclei in this particular staining technique, because most of these nuclei of the neurons are highly active, therefore euchromatic in staining pattern. The similar pattern can be appreciated in the cerebellum. This is a sagittal section of the brain, and again, not human. Here's the cerebrum, and here's the cerebellum that is cut sagittally. And here again, the cortex is comprised of the gray matter, and the medulla, the inner layer, is comprised of these elaborate branching white matter, which is collectively called the arbor vitae, which translates to the tree of life, which is quite poetic and it's quite beautiful. The right and left cerebellar hemispheres too contain the nuclei within the medulla, which is not shown in this mid-sagittal plane. And all of these white matter traversing within the medulla, forming this beautiful branching pattern, are the tracts, which again is forming the arbor vitae. Looking at the gross anatomy of the cerebellum at the histological level, we can still appreciate number of these smaller gyri with undulating outer layer of the gray matter, which forms the cortex of the cerebellum. And the white matter can be appreciated right here, forming the tracts. The cortical layer of the cerebellum at the histological level is quite distinctive in that this cortical layer is comprised of 3 distinct cell populations that make up the molecular layer, which is the outermost layer of the cerebellar cortex, containing numerous neurons and their cell bodies, and then the Purkinje cell layer, which are recognized by number of these larger cell bodies with incredible amount of cellular extensions that go in and out of the molecular layer and the layer underneath them. So these Purkinje cells are highly specialized interneurons of the cerebellum. And then the innermost layer of the cerebellar cortex is called the granular layer, and this layer contains the smallest of neuron cell bodies in the nervous system. So each one of these heterochromatic dots belong to the smallest of the neuron cell bodies. Going down the brain, we now have the medulla oblongata. This structure is also known as the brain stem, and as the name suggests, this structure is located anterior but in the inferiormost aspect of the brain. It is comprised of a mixture of white matter tracts and gray matter nuclei, and making a cross-section through a medulla oblongata and looking at it, here we have the anterior side and here we have the posterior side, and in fact, we've cut a little bit of that medulla oblongata that is forming the anterior wall of this ventricular space. So this V or U-shaped structure or space is the 4th ventricle. The ventricle is filled with the cerebrospinal fluid and it is lined by the ependymal cells. So if we were to look at a zoomed-in view of the ventricular wall, we'd be expecting to see the ependymal cells lining this space. And also, a lot of these granular-looking structures that are projecting into the 4th ventricles are the choroid plexus, which is lined on the outside by the ependymal cells containing the core of loose connective tissue that contain a lot of capillary networks that's lacking the blood-brain barrier so that the ependymal cells can filter and make the cerebrospinal fluid. We'll look at these structures in a lot more detail when we get into the lab portion. Let's move on to the spinal cord, still a part of the central nervous system that has now emerged out of the skull and is traveling within the vertebral column. Here's a transverse section through a spinal cord. This is the anterior side and this is the posterior side. And from the macroscopic view, we can see that the outer component of this spinal cord is actually looking a little lighter staining than compared to this almost butterfly-looking structure that's forming the interior of the spinal cord. So the cortex, or this outer layer of the spinal cord, is comprised of the white matter, which is the opposite from the brain tissue, and the medulla, this butterfly-looking inner component of the spinal cord, is actually made up of the gray matter, which means we're actually expecting to see a lot of neuron cell bodies within the medulla of the spinal cord and axon fibers that are myelinated in the cortical layer of the spinal cord. Let's see if that is the case. We'll sample this small region and look at it under a higher magnification. And lo and behold, we're seeing a nice margin between the cortex and the medulla, the cortex on the outside and medulla in the inside. We can see that in the medulla, we're seeing a lot of these darkly staining and large cell bodies of the neurons. And within the cortex, we're seeing a lot of these really tiny dots, which are the cross-sections of the axons surrounded by these clear staining circles, which are indicative of the myelin sheaths that are provided by the oligodendrocytes. This anterior aspect of the medulla is called the ventral horn, or also known as the anterior horn. And the neuron cell bodies that are found here are primarily the motor neurons, which means these are all multipolar neurons with lots of dendrites, and they're axons that emerge out of the medulla and travel outside of the spinal cord to become a peripheral nerve. And such motor axon will go all the way down to their effector organ, which happens to be a lot of skeletal muscles in distant sites. The posterior component of the medulla is called the dorsal horn. And the neuron cell bodies that are found here typically function as the interneurons because there are sensory neurons positioned outside of the spinal cord, which bring in the proximal fibers through the white matter into the gray matter in the medulla to synapse with the dendrites of the interneuron, which then is relayed by the interneuron axons with some other neurons, either above or below this level, or sometimes this axon may go directly into the ventral horn and synapse with the dendrites of the motor neuron. Central canal is a central opening in the middle of the spinal cord. It's a really tiny opening that's filled with a really small amount of cerebrospinal fluid, and it is also lined by the ependymal cells. And in this image, we're actually presented with a really good opportunity to look at the supporting tissues of the central nervous system. So we'll focus our attention to the boxed area there and zoom in on it. And at this higher magnification image, we are able to see the cortical layer here, which is comprised of the white matter, aka a bunch of these axons that are cut in a transverse section that are also myelinated by lots of these oligodendrocytes, which are showing up as these clear circles outside of the axon. Covering the outside of the cortex, we have these protective structures of the CNS, collectively called the meninges. The outermost layer of this meninges is the dura mater. It literally translates to a tough mother. So this outermost layer, comprised of the dense irregular connective tissue, is the dura mater, which encases all the cerebrospinal fluid that's bathing the outside of the brain and the spinal cord. Deep to the dura mater, we have the arachnoid mater. Arachnoid refers to the spider web-like. This is a loose connective tissue under the dura mater that has a lot of webbings that go between the dura mater and the innermost layer of the meninges. And it is within this webbings of arachnoid mater, called the subarachnoid space, we'll find the cerebrospinal fluid as well as a lot of vasculature that travel through this space. And lastly, we have the pia mater. This is the most delicate and thinnest layer of the meninges, and it sits just on top of the neural tissue. This tiny little layer follows every single contour of the gyri in the brain and little folds within the spinal cord. So when we hear terms like the epidural or subdural hematoma or procedures, we're talking about bleeding or introduction of anesthesia above epi, the dura mater, or below or sub dura mater. With the same image, we can also appreciate some PNS neurohistology and its correlation to the neuroanatomy. So here is the central nervous system, or the spinal cord. Everything that is outside, even the axon extensions that are emerging from the same neuron, whose axon travels within the spinal cord, as soon as it emerges outside of the spinal cord, it then becomes the PNS, while the axon that is traversing through the spinal cord is still considered a part of the CNS. I know that's weird. Another thing that's weird is that the portion of the axon that is traveling within the CNS is myelinated by the oligodendrocytes, but as soon as it emerges from the spinal cord, it is now myelinated by the Schwann cells. The most proximal components of the peripheral nervous system, meaning the closest to the spinal cord, are the ventral and dorsal roots. The ventral roots are collections of motor axons that are emerging from the ventral horn or the anterior horn of the spinal cord. And the dorsal roots are the collections of axon fibers that are traversing from a structure called the dorsal root ganglia and are plugging into the dorsal horn or the posterior horns of the spinal cord. So while the ventral roots carry the motor axons, the dorsal roots carry the sensory axons. And the sensory axon fibers actually emerge from the unipolar axons whose cell bodies are located or concentrated within this anatomic structure called the dorsal root ganglia. Looking at this dorsal root at a higher magnification, we can appreciate this bundle of axons that are traveling together and are myelinated at regular intervals by the Schwann cells, with nodes of Ranvier here and there quite readily visible. And a lot of these elongated nuclei belong to those of the Schwann cells. Looking at the dorsal root ganglion histology a little more closely, and again, this is where a collection of unipolar sensory neuron cell bodies are aggregated. So looking at the dorsal root ganglion at a higher magnification, we can appreciate these large neuron cell bodies with large euchromatic nuclei and punctate nucleoli that are highly concentrated in this organ. At even higher magnification, what we can appreciate now are not only the neuron cell bodies of the unipolar neurons, but also we can see the speckled appearance of the cytoplasm, which are the Nissl bodies, the aggregates of rough ER. Surrounding each one of these neuron cell bodies are these smaller and round nuclei, and again, these satellite cells are the glial cells in the PNS that are kind of an equivalent of the astrocytes in the central nervous system. In the vicinity within the dorsal root ganglion, or the spinal ganglion is another word for one of these, we're still able to see bundles of these axons that are traversing through this structure that are still quite heavily myelinated as well. Once these fibers emerge from the dorsal root ganglion distally, the sensory fibers will soon emerge with the ventral roots. And together, these fibers will comprise a spinal nerve, which contains both the motor and sensory axon fibers. A typical nerve then is comprised of numerous motor and sensory axon fibers that travel together. And as these axon fibers are traveling together, they're sometimes organized into smaller bundles of axons called the fascicles, then can be referred to as the nerve. Here's a longitudinal section through a nerve in which we can actually see a few fascicles here that are traveling together to form a nerve. And another thing that is apparent here are the abundance of connective tissues that are subdividing the fascicles away from each other, as well as these denser looking connective tissues that are bundling a group of fascicles together. So when we make a transverse section through one of these nerves, we can see that organization of the fascicles and nerves with a greater detail. So in a transverse section through a nerve, we can appreciate that a nerve fascicle or a nerve is actually looking quite cellular, but each cross-section of the nerve and perhaps their surrounding myelin sheets are separated away from each other by a good amount of extracellular matrix as well. And furthermore, the fascicles themselves are separated by a thin layer of connective tissues, and then bundles of these fascicles are then surrounded by this much thicker, dense, irregular connective tissue component. So a lot of these connective tissue and extracellular components comprise the supporting tissues of the nerve in the PNS. The outermost layer that is made up of this dense, irregular connective tissue is called the epineurium. It also brings in a lot of vasculature to support the neural tissues. Then we have the perineurium, which are a collections of dense, irregular to loose connective tissue that separate the fascicles of axon fibers away from each other, and again delivering smaller branches of vasculature into the neural tissue. And then a very fine and loose connective tissue to reticular connective tissue type of networks are what forms these clear looking extracellular structures outside of each axon and their surrounding myelin sheets. These are the endoneurium. At a higher magnification, we can really appreciate what is going on here. Here's a single axon cross section with the surrounding myelin sheet. sheath, and outside of that we have this non-staining area, which is that loose connective tissue to reticular connective tissue network, which forms the endoneurium. And kind of partially separating this axon fascicle from this axon fascicle is this connective tissue element, which forms the perineurium, and outside of that we would have the epineurium right here. Another thing that's worth noting about the nerves is the fact that some axons that travel within the nerve are myelinated, but there are also some fibers that are not myelinated, which are called the non-myelinated axons. So when we look at a higher magnification of this longitudinally cut section through a nerve, in which we can see a clear separation of the axons that are surrounded by this clear Schwann cell myelin sheaths. But there are also regions within this nerve where that staining pattern is a little more dense. And that's oftentimes because these regions contain the axons that are not myelinated. So there's higher concentration of not only the axons, but also some Schwann cells. So what's happening with these non-myelinated axons is that although they are not properly wrapped, is that when we look at the cross-section through this area, is that we do have Schwann cells with elongated nuclei and their cytoplasm that are extending out. But these Schwann cells, instead of wrapping themselves around a single axon, they have enormous amount of little branches that surround number of axons that are traveling through in the vicinity like this. So the axons are still protected by the Schwann cells and their microenvironments regulated. All right, let's end with a discussion about the axon injury and its repair. Here's a normal motor neuron that is innervating a muscle fiber. Let's say that unfortunately that there was an axon injury at this location. Within about 2 weeks of that axon injury, we'll see a couple of changes. First of all, we'll see a swelling or edema of the neuron cell body, and number of these Nissl bodies will degrade away. So what usually appears as the basophilic speckles within the cytoplasm of the neuron cell body will start to disappear. We're essentially losing color. So from this perspective, we call the loss of Nissl bodies the chromatolysis. Chromo referring to the color and lysis referring to the death or degradation. So we're losing the color. Again, this is a generic term referring to the loss of color under histological observation. Cytologically, chromatolysis results from, again, the degradation of the Nissl bodies. Now, distal to the injured sites, the axon fiber will degenerate as well as the Schwann cell wrappings as well. And what starts to happen to the effector organ, in this case the skeletal muscle, is that this muscle will start to atrophy. The muscle will not die as long as it has vascular supply, but without the proper neural input, the muscle will start to lose its volume. In the next week or so, the proximal end of the axon will start to grow little branches, which is indicated over here. And there will be proliferation of Schwann cells that'll form a solid cord of Schwann cell column that is still attached to the effector organ. And at this point, if one of these branches of the proximal axon happens to find this Schwann cell column, lo and behold, this axon fiber will continue to grow down the length of this cellular column and re-innervate the end effector organ, in this case the muscle. The Schwann cells will eventually reorganize themselves to form nice little Schwann cell wrappings with proper nodes of Ranvier, and with the re-established neural impulse, the effector organ, or the skeletal muscle in this case, will become activated and regain its bulk or volume. And this type of repair process takes approximately 3 months, if not longer. And unfortunately, should one of these axon branches from the proximal region of the injury not find the Schwann cell column, it'll eventually lead to the death of the Schwann cell column and inability to ever re-innervate the effector organ. So this is the basis behind a limited ability of the neural tissue to repair the axon injury. Here's the histological image of the neural tissue with several neural cell bodies with chromatolysis, highlighted by the swelling of the neural cell body as well as the loss of the Nissl bodies.
Thank you for watching this video from the Da Vinci Academy Histology video course. which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.